Welcome to the unit on prokaryotic cell biology. Throughout this unit, we're going to talk about the various structures and functions that we find in prokaryotic cells. We'll also look a little bit at the difference between bacteria and archaea, two different domains in the prokaryotic classification. Okay, let's go ahead and get started with a brief introduction. The two main classifications of cells, prokaryotic and eukaryotic, should be familiar to you from past classes. Prokaryotic cells, those without a membrane-bound organelles, were the first living organisms to evolve on the planet about 3 billion years ago. They actually had the planet all to themselves for about 2 billion years before the first eukaryotic cells arose. These early prokaryotes gave rise to all life on the planet, with bacteria evolving first, shown here in the overall domain family tree in green, followed by the archaea evolving next from the main common ancestor, and then as we continue down the line, we get to the evolution of the eukarya. Those three domains of life are based on ribosomal RNA sequencing, and the field of microbiology covers all three. In this video, we're going to be talking about the first two, as both bacteria and archaea are prokaryotic cells. You should remember from past classes that prokaryotes are small unicellular organisms that lack a nucleus and other membrane-bound organelles. However, hopefully this image here makes you realize that just because they don't have membrane-bound organelles, there's still a lot of stuff and a lot of structure going on inside a prokaryotic cell. We can see DNA spread throughout here. There are ribosomes and lots of different proteins throughout the cytoplasm. We're going to talk about some of those internal structures throughout this unit. The prokaryotic cells include all cells within the bacteria and archaea domains, which makes them the most widely distributed and most abundant organisms on the planet. There are an estimated 1 billion different bacterial species found on the planet. And remember, we don't know how many archaea existed until the 70s. Which means we still don't have enough information to understand how many of them might be on this planet and make a reliable estimate of their species. So bacteria are just labeled as to be determined. While prokaryotes may all be small, single-celled organisms, there's actually a wide diversity throughout them. This image shows a phylogenetic tree of the bacteria domain, which highlights some of these differences between bacterial species. These thermophiles, for example, live at temperatures close to boiling that would kill both you and all these other bacteria on the list, while cyanobacteria contain chlorophyll and are able to do photosynthesis, something that most of these other bacteria listed here could do. This highlights some of the diversity of bacteria. In fact, some bacterial species are more different from each other than you are from a plant cell. So don't let the fact that they are all single cells fool you into thinking they are all the same. Throughout this unit, we'll be looking at the various shapes and structures found within bacterial species. The information is important not just because those structures provide the variety of functionality that we see between bacterial species, but also because they are used in the medical field for diagnosis. You can classify bacteria based on their shape and other features. Based on those classifications, we can begin to identify a bacterial species just by observing it under the microscope. For example, if you took a wound swab and looked at it under a microscope and you identified gram-positive, cocci-shaped cells that formed clusters, then there's a good chance you are looking at Staphylococcus aureus, more commonly known as a staph infection. As we go through this unit, the terms on this slide will become more familiar to you. While all prokaryotes are single-cellular, they are not all the same shape. Additionally, sometimes those single cells associate with one another to form characteristic cell arrangements. The two most common bacterial shapes are cocci and bacilli. Cocci forming round spherical cells, and bacilli being elongated rod-shaped cells. Looking at cell arrangements, you'll also notice that some of these terms you may recognize as genera of bacteria you know. We've already mentioned Staphylococcus aureus, which you may know as the staph infection. The prefix staph actually means clusters, and cocci we just said were round bacteria. So the genus name Staphylococcus actually means clusters of cocci or spherical bacteria. Under a microscope, they actually tend to look like grapes on a cluster. Similarly, the prefix strepto means chains. So the genus Streptococcus defines species found as long chains of round bacteria. All cells, no matter if they are prokaryotic or eukaryotic, contain a cell membrane that functions to separate the cytoplasm from the external environment. It controls what enters and exits the cell through both diffusion and active transport. All cell membranes are made up of a lipid layer embedded with proteins that function in the transport of molecules across the membrane, as well as receptors and enzymes for various cellular processes. Interestingly, the cell membrane composition of bacteria is similar to eukaryotes, but is very different than the archaea. In this course, we're not going to talk about the archaea very much, typically because while few archaeal species have been found associated with the human body, they are in the vast minority compared to bacteria, and there are no currently known archaeal pathogens for humans. However, I do want you to realize that just because they are both prokaryotes, there are key differences between cells in the domain archaea and those in bacteria domain. The structure of the membrane of phospholipids is one of the biggest differences. Bacteria and eukaryotes have a lipid bilayer that you've learned about in past classes. They contain phospholipids, shown here, that have a glycerol with a phosphate head group attached and two fatty acid tails. Those fatty acid tails are attached with an ester linkage. This is different than the archaea. Archaea will contain glycerol and a phosphate, but the attachment for their fatty acids is a different chemistry, with these being ether linkages. Even more interesting, some of the fatty acid tails in archaea can actually be branching, which doesn't exist in bacteria or eukaryotes. When you look at the membrane structure itself, both bacteria and archaea always have the characteristic lipid bilayer with one layer of fatty acid tails associated with the other layer. However, some archaea actually have monolayers of a membrane where the phosphate groups on the end are actually part of one molecule as opposed to two molecules. Archaea are also well known for being extremophiles, which is because they are found in extreme environments, such as the hot springs in Yellowstone or deep under the polar ice caps. However, they're also found in normal locations, such as the soil, and we now know there are at least a couple species associated with the human microbiome. Interestingly, archaea are actually more closely related to eukaryotes than to bacteria when you examine their gene expression machinery.